Hi, hello. This is Dr. Jagadish Narendra Prasad, Senior Lecturer in Botany, Sri Chaitanya Goshala, Vijayawada. Today, let us discuss about amino acids. First, we'll go with amino acids, then we'll discuss some details regarding the proteins. Amino acids are organic compounds with both NH2 and COOH. Say amino group and carboxylic group attached to the same carbon, that is alpha carbon. Therefore, amino acids are called alpha amino acids because both amino group and carboxylic group attached to the same carbon. As we know, the carbon valency is 4. Therefore, the 4 valency of carbon is substituted by 4 functional groups. Therefore, amino acids are also called substituted methanes. Let us see how exactly the structure of amino acid exists. So, there is one NH2 amino group, there is one carboxylic group, there is one hydrogen, and there is a variable group, otherwise called R functional group. So, the 4 valency of carbon is substituted by the 4 groups. Therefore, they are called substituted methanes. Depending upon the nature of our group, many types of amino acids have been identified, out of which only 20 types of amino acids are present in the proteins. Proteins contain only 20 types of amino acids, even though many types, almost more than 700 types of amino acids are present in the living tissues. Out of this, only 20 types are present in the proteins. So, these are called proteinaceous amino acids. Say, if the R group is hydrogen, then it is glycine. If it is methyl, then it is alanine. If it is hydroxymethyl, then it is serine. These are the 3 important amino acids out of 20 types of amino acids present in the proteins. In the same way, depending upon number of carboxylic groups and amino groups present in an amino acids are again classified into 3 types: acidic amino acids, basic amino acids, and neutral amino acids. Acidic amino acid is nothing but an amino acid with a 2 carboxylic group and 1 amino group is acidic. The best example, glutamic acid, aspartic acid are the best example for acidic amino acids. Basic amino acids are those with 2 amino groups and 1 carboxylic group. Example, lysine, arginine, these 2 are the best example for basic amino acids. I am discussing about only proteinaceous amino acids here. Out of the 20 proteinaceous amino acids, 2 are basic amino acids and 2 are acidic amino acids. And neutral amino acids are those where 1 carboxylic and 1 amino group will be present. Example, valine, alanine, glycine, serine also, serine. These are all some important examples. In addition to this, apart from these 3, there is also aromatic amino acids in the living tissues. Aromatic amino acids in the sense, these are the amino acids with benzene groups, simply, in a simple way. Example, tyrosine, phenylalanine, tryptophan. These are the three important examples for aromatic amino acids. And another most important thing is a particular property of amino acids is ionizable nature of NH2 group and COOH group. The NH2 group and COOH group present in the amino acids, this NH2 group and COOH group present in the amino acid have the ability to undergo ionization. Therefore, in solutions of different pH, the structure of amino acids changes. For example, if an amino acid is kept in acidic solution, say pH less than 7, then you will be getting this kind of structure. It is a fully proteinated form. Whereas, if the same amino acid is placed in basic solutions, neutral solution, say pH 7, then you will be getting this kind of structure. Here, this is acidic and this is basic. So, this form of amino acid is consisting both acidic group and basic group. Therefore, it is known as Zwitter ionic form. Whereas, if this amino acid is placed in a medium with basic solutions, then you will be getting a deprotonated form where amino acid get basic structure. This you can see at a pH greater than 7. Like this, in solutions of different pH, the structure of amino acid changes because of ionization ability of NH3 and the COOH group. NH3 and COOH group. So, this is what you can see with regard to the structural details of amino acids. So, as we understood now that amino acids are building blocks of proteins. A protein is composed of many amino acids. Proteins are heteropolymers. Polymers are of two kinds, homopolymers, heteropolymers. Homopolymer is one with only one kind of repeated unit, and heteropolymer is one with more than one kind of repeated units. As proteins are made with different types of amino acids, like around 20 types of amino acids are present, therefore, they are said to be heteropolymers. No protein in the living tissues is a homopolymer. This we have to remember. Homopolymer, I will just give you a simple example. If a protein is made with only tryptophan, it is homopolymer. A protein with tryptophan, lysine, alanine, glycine, etc., heteropolymer. Never these proteins are present in the living tissues in the form of homopolymer. They are always in the heteropolymer form. These proteins are again of two kinds, like essential, these proteins have amino acids, and amino acids are of two kinds, essential amino acids, non-essential amino acids. Essential amino acids are those which our body cannot synthesize. Therefore, we need to take these type of amino acids through dietary protein, and non-essential amino acids which our body can synthesize by its own, no need to take from outside or outside sources. And these proteins are doing different kinds of functions in our body. Say for example, collagen is one kind of protein. It acts as an intercellular ground substance. Collagen, the most important thing, collagen is the abundant protein in the animal tissues. Abundant protein in the animal tissues. Whereas in the biosphere, the abundant protein is rubisco. What is rubisco? Rubisco is ribulose 1,5 bisphospho carboxylase oxygenase. It is an enzyme for photosynthesis. C3 cycle enzyme. So this is most abundant protein in the biosphere. So some proteins or enzymes, trypsin, pepsin, rubisco, etc. And some proteins or hormones, say for example insulin. Some are fighting against infectious agents like antibodies. Some are acting like receptors, and some also helps in the transportation of substances, say for example GLUT4. GLUT4 is one transporter that enables glucose transportation into the cells. So like this, the proteins have different kinds of functions in our body. If you look into the structural details of protein, the structure of any molecule means different things in different context. For example, an inorganic chemist explains the structure in a simple way by giving a molecular formula, say NaCl, MgCl2, etc., etc. Whereas organic chemist gives a structure, any structure of any molecule in a two-dimensional view. A physicist conjure up the structure in three-dimensional view. Whereas biologists have provided the structure of a protein at 4 levels. What are they? Like primary structure, secondary structure, tertiary structure, and quaternary structure. Let us see first about primary structure of a protein. Primary structure of a protein gives positional information about that protein. Positional information in the sense, it explains about which is the first amino acid, which is the second, third, fourth, so on. Such information is obtained from the primary structure of a protein. If you depict the primary structure in the form of a line diagram, the left end is called N terminus. It is called N terminus. Whereas the right end is called C terminus. Here N represents that there is a free amino group at the left end of the amino acid. Left end of this protein thread. The amino acid present in this protein, the first amino acid, it is otherwise called first amino acid. In the first amino acid, there is a free NH2 group, so it is called N terminus. In the last amino acid, there is a free COOH group, therefore it is called C terminus. There is a free COOH group, hence it is called C terminus. Now this protein will give you the positional information. This primary structure is again forms into two kinds of secondary structures. One is alpha helix and other one is beta sheet or beta pleated. These are the two different forms of the primary structure. Let us take what is the difference between these two first. Alpha helix and beta sheet have got a simple difference that in alpha helix only one protein thread is got folded. Only one protein thread is got folded. Whereas in beta pleated or beta sheet, more than one protein threads are present. In the alpha helix, intrapeptide hydrogen bonds are present, not inter. Intrapeptide hydrogen bonds between CO group and NH group. Intrapeptide hydrogen bonds. Whereas in beta pleated, there is interpeptide hydrogen bonds are present between CO group and NH group. That is the difference between alpha helix and beta sheet. That means this primary structure does not exist as a rigid rod throughout its length. It is folded in the form of helices. Only some portions of the helix, some portions of the thread is formed into helices. This other regions of the protein which is folded in other form is nothing but alpha helix. So in the alpha helix, what kind of hydrogen bonds are present? It is intrapeptide hydrogen bonds. Intrapeptide hydrogen bonds between CO and NH groups. Whereas in beta sheet, interpeptide hydrogen bonds are present. Interpeptide hydrogen bonds means hydrogen bonds are formed between two peptide chains, because beta sheet itself is made with more than one peptide chains, like 2 or 3 or 4, sometimes many peptide chains will be present. So that is the main difference between alpha helix and beta sheet. This alpha helix is again folded, refolded and forms into hollow woolen ball shaped structure or a spear. This spear itself is called tertiary structure. So tertiary structure is one that appears like a spear. The alpha helix itself is folded, refolded and forms into hollow woolen ball shaped structure. That itself is called as tertiary structure. This tertiary structure is absolutely necessary to carry out various biological functions. It is not that only tertiary structure is essential. Secondary structure also have some biological functions. Tertiary structure is also having, quaternary structure can also do the biological functions. But most of the biological functions in our body are under the control of tertiary structure of a protein. Because the tertiary structure, like most of the enzymes are also in the tertiary form. This tertiary structure results in the formation of active sites in the enzymes. Therefore, tertiary structure is essential to carry out various biological functions. This is what you can see with regard to the tertiary structure. When it comes to quaternary structure, more than one peptide chain, means 2 to many peptide chains, have arranged in the form of a string of spears. String of spears means a chain of spears like this, or in the form of a cube or plate, one upon each other like this. This kind of architecture of a protein itself is called quaternary structure. Quaternary structure. So quaternary structure is composed of more than one type of polypeptide chains. Let us take once again from the primary structure to quaternary structure. Look at here. Now this is primary structure of a protein. Here you are getting the positional information about different amino acids present in this protein chain. Alanine, glycine, serine, valine, leucine, lysine, glycine, valine, etc. This primary structure is folded into alpha helix. And we have to remember that this proteins forms only right-handed helices, never they form left-handed helices. So this is a right-handed helix, alpha helix. This alpha helix is again folded and refolded to form a spear. That spear, otherwise called a hollow woolen ball shaped structure, this itself is called tertiary structure. Many tertiary structures, like more than one tertiary structures, that may be 2 or 3 or 4 or still more, many tertiary structures may have arranged in the form of a string of spears or in the form of a cube or plate, one upon each other, and gives the quaternary structure. One of the best example for the quaternary structure is human hemoglobin. Human hemoglobin is composed of 4 types of polypeptide chains. In that, 2 types are identical. There are 2 alpha type of proteins and 2 beta type of proteins. So this 4 forms into human hemoglobin. That's very important. So this is what you can see with regard to the structural details of a protein. So proteins are heteropolymers. The building blocks of the proteins is a amino acid, and amino acids linked by peptide bonds. When this primary structure of a protein is folded, you will be getting hydrogen bonds. When secondary and that leads to secondary structure is refolded and forms a tertiary structure. In the tertiary structure, apart from peptide bonds and hydrogen bonds, you can also see disulfide bridges, covalent bonds, including ionic interaction, hydrophobic, hydrophilic interactions, van der Waals interactions, all such kind of interactions you can see this. And finally, in some cases, more than one tertiary structures forms into a quaternary structure. Remember, quaternary structure is made with more than one tertiary structure. This is what you can see with regard to the brief discussion about a protein and amino acids. Thank you very much. We'll meet in other class with other topic. Thank you very much.